find within blood. As we talked on Monday, they all start out with one progenitor cell, stem cell, and that stem cell can go down either the myeloid lineage or the lymphoid lineage to give us ultimately the 5 types of white blood cells, platelets, or red blood cells. That process of developing blood cells is called hematopoiesis. They all originate in red bone marrow. They start out as hemocytoblasts, or what I more commonly refer to them as hematopoietic stem cells. Those stem cells then give rise to more stem cells as well as specialized stem cells, which continue to differentiate in response to growth factors, lymphoid stem cells, or myeloid stem cells. The lymphoid ones give rise to the lymphocytes, either the B or the T, while the myeloid stem cells give rise to the other types, the red blood cells, the other types of white blood cells, and the platelets. So if we start with red blood cells, or RBCs, which are also called erythrocytes, they have a biconcave shape. They're like a donut that doesn't have the hole go all the way through. They are about 1/3 of volume of hemoglobin. Some of that is oxyhemoglobin carrying oxygen, and some is deoxyhemoglobin without oxygen on it. They do not have a nucleus. They do not have a mitochondria, so they can't use the oxygen they are carrying. They are just using, just carrying that oxygen. They do produce ATP, but they do it through the anaerobic process of glycolysis, splitting glucose in two, and then going through fermentation with a pyruvate product. So the red blood cell count is the typical amount of red blood cells you would have. It's a useful diagnostic for disease and evaluating the progression of diseases. Changes in the red blood cell count would reflect the oxygen carrying capacity of the blood. I know this slide is kind of messed up as far as the math goes, so check your book to find the correct red blood cell counts. Erythropoiesis is specifically the formation of red blood cells. It occurs complete within the red bone marrow in response to low blood oxygen. That would lead kidneys to release erythropoietin, a hormone that stimulates red blood cell production that works on a negative feedback mechanism. So you would go from the hemocytoblast to the erythroblast to a reticulocyte, and from a reticulocyte to an erythrocyte. And they would appear quite readily in the blood. There are some dietary factors that will affect red blood cell production. Vitamin B12 and folic acid are both required for DNA synthesis, and they are needed for the growth and division of all cells. So although red blood cells don't reproduce, the stem cells to make them need to reproduce and need that DNA synthesis. Iron is required for the hemoglobin structure. Anemia is a condition in which the oxygen carrying capacity of the blood is reduced, and that could be due to a number of factors. It could be a deficiency of red blood cells, it could be a deficiency of hemoglobin. So we talked about some of these on Monday. Aplastic anemia is damage to the bone marrow. Hemolytic anemia, red blood cells are destroyed too quickly. Iron deficiency, you have hemoglobin deficiency. Pernicious anemia is an inability to absorb vitamin B12, and that's usually due to a lack of intrinsic factor in the digestive system. Sickle cell disease we talked about, and thalassemias we talked about on Monday. The major events in getting rid of red blood cells circulate around the issue of conserving the iron. So as red blood cells make their way through capillaries, they ultimately will be damaged in the process. Remember, they're a little bigger than the smallest of the capillaries. Macrophages in the spleen and the liver will phagocytize those damaged red blood cells. The hemoglobin will be decomposed into the two components, heme and globin. The heme will decompose into the iron and a substance called biliverdin. The iron would be stored in the liver on ferritin or moved to the liver on transferrin. The biliverdin is converted into bilirubin, and they are secreted into the bile as bile pigments. The globin is broken down into individual amino acids, and those amino acids can be used to build other proteins. So the small intestines absorb the nutrients. Blood transports the absorbed nutrients. Red blood cells produced in the red bone marrow. Red blood cells circulate in the bloodstream. They have a lifespan of approximately 120 days, at which time macrophages begin to phagocytize and break down the old ones. The hepatocytes break down the hemoglobin. The iron is recycled in the bloodstream. And the bile makes its way to the small intestines. So from the heme group, we make our way into biliverdin with free iron. The biliverdin can be converted into bilirubin. Both of these will serve as bile pigments. Let's talk about white blood cells. The white blood cells are the leukocytes. They're all involved in protecting us against disease. Leukocytes also have a limited lifespan, so they are constantly being replaced. White blood cells are mitotic cells in most cases. They are true cells with organelles. White blood cells are produced also in the red bone marrow. They are under the control of hormone or hormone-like substances known as interleukins or colony-stimulating factors. There are 5 white blood cell classes in 2 categories. They're either granulocytes, because they have small granules in their cytoplasm, or agranulocytes, which don't have noticeable granules in their cytoplasm. The granulocytes were the neutrophils, the eosinophils, and the basophils, and the agranulocytes were the lymphocytes and the monocytes. The neutrophils are also called polymorphonuclear cells, or PMNs. They're small, they have light purple granules. When they're stained with right stain, they have both acid and base components, which is where the neutral came from. Their nucleus is lobed, usually has between 2 and 5 sections. Again, they are also called PMNs, or polymorphonuclear cells. They are one of the first cells to arrive at an infection site. They are very good at phagocytosis, very good at getting rid of bacteria. They make up about half, 54 to 62% of the leukocytes. Eosinophils are coarse, and their granules stain a deep red because they are acidic. They generally have a bilobe nucleus and are involved in allergic reactions. Their original purpose was likely to defend against parasitic worm infestations. Parasitic worms are too big to be phagocytized, so eosinophils dump their products on phagocytic worms, or parasitic worms, in an attempt to kill them. And there's some speculation that because we don't deal in the modern world, or in the developed world, with parasitic infections, that our eosinophils maybe are a little overactive. Basophils also have large granules, but they stain blue. They pick up the basic stain. The granules can obscure the view of the nucleus. They release histamine. Histamine is a powerful inflammation-stimulating chemical. In fact, most of the medications you take to treat a cold during the course of a typical year would be antihistamines, so taking care of undoing what this histamine is doing to your body, not directly attacking the pathogen. They also release heparin, which slows blood clotting. They are very rare, only about less than 1% of the leukocytes. They are very similar in size and shape to the eosinophil. Monocytes are the largest of the white blood cells. They are round, spherical. They have a kidney-shaped, oval, or lobed nucleus, kidney-shaped most typically. They are agranulocytes. They leave the bloodstream, go into the tissues, at which time they mature to become macrophages. They can live for weeks to months. They're very good at phagocytizing bacteria, dead cells, and debris. Those macrophages will become important for getting an immune response going when we get to that chapter. And they come in following the neutrophils. Lymphocytes are the smallest of the white blood cells. They have a large spherical nucleus with a very thin layer of cytoplasm. Again, they are agranulocytes. They come in two major types: T cells, which are involved in cellular immunity, and B cells, which are involved in antibody-mediated immunity. That's what we'll say for now. We'll get into much more detail about what they do in immunity later on in the text. B cells and T cells live for many years. Hopefully we get immunological memory. So diapedesis is the ability of a white blood cell to squeeze between the cells of the capillaries and leave the blood vessel, migrate towards an infection site or an insult of some type. To do that, they use cell adhesion molecules that help the cell find out where it's going. When they get to a target, many of them are able to do phagocytosis. We talked about neutrophils and macrophages, monocytes. That would be to engulf and digest a pathogen. The inflammatory response is a reaction that restricts the spread of infection. It's promoted by basophils, which secrete heparin and histamine, and it involves all those cardinal signs of inflammation, including increased capillary permeability, which can lead to swelling. And to get where they are going, white blood cells use a process called positive chemotaxis, which means they follow trails of chemicals to their source. So they go towards these chemicals. Positive chemotaxis. So in this instance, we have a splinter that has punctured the epidermis. There's bacteria here. Even if this was a sterile toothpick, you would still have chemicals coming from the injury itself. Those bacterial chemicals and injury chemicals are slowly diffusing from that site. And our white blood cells will follow the trail of those chemicals from where they are weakest to where they are more concentrated until they make their way to that site of injury. And the first ones there would typically be neutrophils. If neutrophils don't take care of the problem, they'll be followed by monocytes and macrophages, and those macrophages would then activate the immune response to help make immunity against whatever had come in on that toothpick. So white blood cell counts are the counts of white blood cells. Again, this slides don't translate very well, so check your book for what an average white blood cell count is. What's important here is that leukocytosis is a high white blood cell count, while leukopenia is a low white blood cell count. High white blood cell counts could mean acute infection, even as vigorous exercise or a loss of body fluids. Whereas leukopenia, low white blood cells, could be some diseases such as typhoid fever, flu, measles, mumps, chickenpox, AIDS in particular, polio, or any other anemia. And what is usually done is a differential white blood cell count, which would give you the percentages of each type of leukocyte. So you don't just have which leukocytes are low. Neutrophils would be up in case of a bacterial infection. Helper T cells, CD4 positive T cells, are greatly diminished in an active HIV infection. So, clinically, leukemia is a cancer of the white blood cells. It can be classified as acute or chronic. We named the leukemia based on which of the two pathways or other pathway it comes from. So lymphoid leukemia would be lymphocytes, myeloid leukemia would be granulocytes. There are very good treatments for some leukemias, particularly the acute ones that tend to show up in children. However, we are still struggling with some of the more chronic ones that tend to show up in adults. Platelets are cytoplasmic fragments of a cell called a megakaryocyte. They produced by hemocytoblasts in response to the hormone thrombopoietin. So the hemocytoblast would go to a megakaryocyte, would mature to a thrombocyte. And the thrombocyte would be the platelet. Platelets do not have a nucleus. They are less than half the size of a red blood cell. Again, the platelet count should be between 150,000 to 350,000, but check that in your book. And they are involved in hemostasis, which is the stopping of bleeding in blood vessels that become damaged. And they do that by sticking to the broken surfaces, releasing serotonin that causes smooth muscles in the walls of the blood cells to contract, begins the process of clotting. So here are our components: red blood cells, also called erythrocytes, white blood cells that are either granulocytes or agranulocytes, the granulocytes being neutrophil, eosinophil, and basophil, agranulocytes being lymphocytes and monocytes, and platelets, which are also known as thrombocytes. All right, we'll stop there.